If or not really. 111. I think we're live. Yeah. Okay, where we stopped. I actually want to know. Oh yeah, we did the no response on which connection to enable during research. God, it's a great research. Let's retry it. The brain actual. What is my researches, right? 1,595 sources in 1 hour. We live in an amazing time. Macrophage agent, this is ready. Is evaluate your or intuition against the 26 state field across history, convoy through mechanism argument why car macrophages are mismatched to suitable targets, including charisma 25 collapse. Okay, so it doesn't work. The reports close with rigorous reformulation of the startup thesis and the explicit assignment of which part of your intuition survived. Interesting. Reality check. What part of this user intuition survived scrutiny? The delusion framing that age elevates factors in all blood as a major driver, and their removal is therapeutic, is well supported by Megdipur 2020 Kim broad yeah, all that fancy, not fancy, like legit researches, right? Of clear and specific pro-aging factors, or they know them. It sounds and has genetics and pharmacological support. The instinct to attack aging in the blood level is consistent with the most reproducible regeneration interventions in mammals. What part oversimplify? Binary young blood has no youth factor, just absence of bad factors. I think no. Oh, okay. Overstated into P PF4 is the real reproducible use factor mechanism. Okay, so basically the young blood also has a good stuff in it. Interesting. The macrophages modality is mechanically misaligned. Not true, because they're eating like pathogen. I was like, oh yeah, just eat this bad stuff, like this one. And maybe they are in different sizes completely. Modality is mechanically with the soluble target use case. This is not a minor engineering hardly, but a reversal of what phagocytes evolved to do in the sector of luxury. What is charisma? It's everywhere charisma, charisma. Just collapse while working on the easiest cellular target case. Is that a startup that's trying to do that? It is. Harnesses the power of macrophages. Damn, they exist. That's awesome. Oh, but they want macrophages to eat cancer. No, maybe not. I'm just guessing, like from this homepage. Okay, I'm wondering if any of the crazy ideas that I'm gonna throw on the wall and see what sticks by connecting random, like almost random similarities in different systems. I wonder if any of those ideas will be legit. But that's fun. I mean, the cool part is that you don't need to spend like hours to do all that research. It's actually done it like in 1 hour. Amazing. Let's go really like into testing a little bit more, get the basics, like see my gaps and so on. Let's go. Let's go. Question 11. Are you ready? New zone. What is the cell membrane made of and how does it work? Ooh, I think membrane made from, again, protein or amino acids. Proteins made from amino acids. So I would guess from the proteins, maybe not. And it's pretty cool because it can allow some stuff to pass through from external world to pass through into internal world, and then but majority stuff is not passing obviously. And in order to pass it, I think it has like special kind of lock and or key or gates that also certain proteins bind it, maybe not proteins, enzymes bind it, and then it's like passing through. I'm just throwing pretty much what I remember from biology 25 years ago, maybe more, 25 years ago. So I don't know what it's made of, but I definitely know that it's isolate insides, organelles, nucleus from outside world, from external. And that book, which is The Self Gene, is the best describing it. And I really like that analogy because they're saying it's actually the gene is an organism and it developed kind of protection for itself and then develop its own how I would say it, starship. So the cell is the starship, right, in external world. And then it's actually even develops the multicellular organism, so it can reproduce and pass the genes further. So the multicellular, it's the same intent or will from the gene to survive and procreate. And VR is just a result of it. Oh, where's my questions? Oops. Did I put V? What's that? And VR is just oh, oh my god, stop it, stop, stop, edit. And VR human. Uh, the voice to text is weird, especially when you seek. Made from protein, no. Plus the self-engine reference. Dow, I don't know how to pronounce his name. I'm sorry. I'm not gonna butcher it. Would approve of colony of starship. Let me score the membrane part. Member of proteins, not quite. Cell membrane are primarily made of lipids. Lipids. Specifically phospholipids. Is the same as phosphates? No, probably not. Lipids or phospholipids. I need to see them. The images. Okay. Leukoprotein. Protein with carbohydrate attached. Okay. Where is lipids? I'm assuming this red stuff is lipids. Phospholipids. Bilayer. Okay. Protein channel. Oh, okay. So this is phosphate or stuff like, right? Interesting. What else is here? Yes, that's the lipid. Phospholipid, one of them. But so it's not protein. Lipids. Phosphate head. Fatty acid tails. Oh, here's the molecular stuff. So this one looks like exactly phosphate for DNA, right? Yeah, that's it. That's a different one. But this one I remember from a DNA. What is DNA made of? DNA. DNA. Yeah, look at this. The base, obviously, deoxyribose, the sugar, and phosphate. And phosphate is O, 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 and H. I forgot H. C, T, A, G. A, T, C, G. That's how I remember. A, C, T, G. And T swap with the U in RNA. That's what I remember from last time, right? I think T swap was a U. A, T, C, G. Yeah, T is swapped for the U. Oh, that's cool. Wow. Double helix. So this is sugar, this is phosphate. Yes. A, T, C, G. A always pairs with T and C always with G. T, A, T, A, H, G. Ah. Major groove, minor. Oh, right, because they're not evenly spaced. I forgot about that. The grooves between double helix. Okay, so but on this phosphate there is no H. Here's the H. Here's no H. Why? This one H. And this one no H again. Phosphate bound. Okay, no H, no H. This one H. Oh, it's a OH. It's not from a phosphate. Oh, I'm getting confused. Yeah, it's exactly the same. The lipids has exactly the same phosphate. Phospholipids bilayer. Hydrolipid head has a phosphate, but this one doesn't have a chemical formula for some reason. Unsaturated fatty acids. Saturated fatty acids. Okay. This actually is the fourth biomolecular category you have been missed. I need to get to know this. The four families again: carbohydrates, lipids, proteins, nucleic acids. Okay. I need to somehow carbohydrates, proteins, lipids, and nucleic acids. AKG sign. Yeah, let's go.
In all living things, there are four important classes of large biological molecules. These molecules are important in chemical processes in the cell, and the structure started. I mentioned there are four classes. Only carbohydrates, proteins get started. I mentioned there are four classes. Only carbohydrates, proteins, and nucleic acids are known as macromolecules, which consist of polymers. Lipids is slightly...
111. I think my mic not connected. I'm sorry. One. Yeah, much better. Sorry.
Slightly different in that they're defined by molecules. Let's get started. I will cover how macromolecules are built and break down the structure and function of all four classes of large biological molecules. Let's get started. I mentioned there are four classes. Only carbohydrates, proteins, and nucleic acids are known as macromolecules, which consist of polymers. Lipids is slightly different in that they're defined by a property rather than by their chemical structure. A polymer is a long molecule consisting of many similar monomers that are linked together by covalent bonds.
Monomers. Macromolecules. Nucleic acids, it's a chain of nucleotides. Protein is a chain of amino acids. Polyhydrate, it's a chain of sugar.
Recall what covalent bonds are: a strong chemical bond in which one or more pair of valence electrons are shared. These repeating units are the building blocks of a polymer, and they are called monomers or subunits. Now, each of these polymers is made up of a different type of monomer, but the way cells make and break down polymers are basically the same. And these processes are facilitated by enzymes, which are specialized macromolecules that speed up chemical reactions. We cover how and why in the enzymes lecture. So,
is it an enzyme only in cells or between cells as well? I think I'm gonna watch this one as well. I need to bench watching this. Oh, biochemistry. Okay, Dave, hit me up.
Hey, it's Professor Dave. Let's talk about biochemistry.
He knows a lot about the science stuff.
What is biochemistry? Is it biology? Is it chemistry? Well, as you might guess, plea of the sciences, biochemistry sits right here. If we go smaller, we get into small molecules made of just a few atoms, which is chemistry. And even smaller than that, we get to the particles that are even smaller than a single atom, and that's particle physics. If instead we go much bigger, We are looking no longer at individual molecules, but entire cells or parts of a cell. That's biology. Right in between is biochemistry. Is it important or general health is a chemical process by which molecules in your body break down the food you eat.
Starch. That's carbohydrates. And they made from sugar, chain of sugar. That's sugar. Polysaccharide. Storage glycogen. Oh, I know glycogen, it's in the muscles.
Your health is governed by biochemistry as well, since all diseases have a molecular basis. When you really get down to it, the domain for completely strategy, opting instead for unsubstantiated things on basic. It's fundamentally the same stuff we learned in general and organics happen. If you are someone that just wants to organic chemistry playlist,
General chemistry, organic chemistry.
That you really will want to watch first in order for any of this to make sense. Everything in this series will be pretty easy to understand. When we see a picture of time, because there are hundreds or even thousands of atoms in it, which would be impractical to draw out individually using the line notation we are familiar with. But we absolutely must be able to understand this enzyme on the molecular level so we can know what functional groups it's made of and how it does basic chemistry. Otherwise, biochemistry seems like magic, but it's not. It's just many surprisingly simple chemical reactions, even if they are applied in complex ways and on huge molecules. My goal with this series is to focus on the basic concepts of biochemistry and illuminate them for the student and the common viewer alike. I may occasionally mention details that aren't that important to non-students, and I also may gloss over some of the more technical details. Hey, it's Professor Dave. Let's talk about carbohydrates. We've all heard of carbohydrates and sugars, especially if we are loading up with energy for the big game or if we're on a diet. But many people don't know that these words refer to the same class of molecules. Carbohydrates got their name because they are literally hydrates of carbon, molecules with several carbon atoms that each bear a hydrogen and a hydroxyl group. When they are small, they are also known as sugars, and their names end in "-ose", like glucose or sucrose, which is the sugar that we commonly refer to as simply sugar. Let's learn about monosaccharides first. These are the monomeric units that polymerize to form polysaccharides. We can name these according to the number of carbon atoms they have, which will be between 3 and 6. So a 3-carbon monosaccharide would be a triose. If it has 4 carbon atoms, it's a tetrose. 5 carbons would be a pentose, and 6 would be a hexose. In addition, all carbohydrates will have either an aldehyde or a ketone function.
Okay, so this is what's the name? Monomer of obviously of poly. Oh, aldose, ketose.
Functional group in the molecule. So we would refer to those as an aldose or a ketose, respectively. Combining these conventions, we could refer to this monosaccharide as an aldohexose and this one as a ketopentose. Each of these carbons, the ones that bear both a hydrogen and a hydroxyl, will be a chiral center.
How can you remember all this?
And the convention for drawing linear monosaccharides is to use Fischer projections. When we look at these, it is important to understand that each vertex is a stereogenic carbon, and we show the hydrogens and hydroxyls on either side. With Fischer projections, horizontal lines are implied to be wedge bonds, and the vertical lines are implied to be dash bonds. To remember this, just imagine a bow tie on each carbon, like it's going to the chemistry prom. Fischer projections are just a different way of visualizing a molecule, which we can see if we draw this molecule in line notation and view.
Oh, that's cool. That's a Fischer projection. That's so cool. I didn't know about it.
View it from the side. The Fischer projection is what we would see if we were in the plane of the screen looking directly at the chiral center. However we choose to view it, it's important to realize that Fischer projections do not imply a flat molecule with 90-degree bond angles. These carbons are still tetrahedral. We just save time by drawing sugars this way. When we draw linear monosaccharides, we always put the aldehyde or ketone at the top and draw the rest of the molecule downwards, and we report the stereochemistry of the molecule by looking at the chiral center at the bottom, furthest away from the carbonyl. If on this carbon, the hydroxyl points right, it's a D sugar. If it points left,
I think it's too deep. It's too deep. We're going back. Oh, it's actually it's phospholipid has a head that loves water, hydrophilic, and two tails that repel water, hydrophobic. Oh, interesting. In water, they spontaneously arrange into a double layer, heads facing outwards towards water on both sides, tails hidden in the middle. This is so cool. This is I just don't understand if the lipids made from molecules and the water are molecules too. How do they? So the water is not really water. I mean, sorry, it is water, but it's not what we use to water, like liquid, right? The water molecule means like H2O. Because when I read water, I'm envisioning the water that we drink, but it's just H2 on a molecular layer. But I like that. I didn't know that the head is hydrophobic. Hydrophilic. And the tails are hydrophobic. Hydrophilic. Hydrophobic.
Hydrophilic.
Mm-hmm.
Hydrophobic.
Yep. This is a phospholipid bilayer. It forms automatically, which is why biologists think polymembranes may have assembled without any design. Oh. The bilayer is only in the middle, so the water soluble stuff, iron, sugar, most molecules, can just pass through. That's your isolation function. Can't just pass through. Okay. Lock and key gate. Correct concept. Proteins are embedded in the membrane, floating in the lipid sea like icebergs. These membrane proteins act as the channels, pumps, and receptors. So the membranes is lipid, but the gates and sensor are proteins. Okay, you had the right players just swapped. Who was the walls and who was the door? Okay. Allowing some stuff through blocks most. Correct. This property is called selective permeability. It's currently the most important property of life without selective bacteria. I want to know how big the cell can be. How big the can be and what is preventing to be even bigger. Diffusion. Great intuition. Diffusion is exactly the main constraint, but it's part of the biggest story about surface area to volume ratio. Yeah, I remember the surface when we talk about mitochondria. Remember, to increase the surface, it's right away pop up. The surface area is important, but actually not the surface area, but surface area to volume ratio. You just simply don't have enough surface area, right? Okay, typical size cells. Most eukaryotic cells. Okay, eukaryotics, that's the ones with the nucleus That's with DNA inside of nucleus. And prokaryotics is the one where DNA was flowing without nucleus. Am I right? I need a companion who just listen to me, and when I when I ask an am I right, it will just tell me yes or no. Okay, most aqueous cells are 10 to 100 micrometers. Bacteria usually 1 to 10 micrometers, but they are wild outliers. Ostrich egg yolk is a single cell, about 15 centimeters across. Wow. The egg is a single cell? Is all eggs a single cell? A chicken as well? No. Is it? Why the organelles is not so big? I'm gonna ask that question. Caulerpa taxifolia, a green alpha, up to 30 centimeter. Technically one giant multinucleate cell. Human nerve cells are axons of a static nerve neuron, can be over 1 meter long. So very thin. So it's interesting. Axons are single cell, almost 1 meter long. Deep sea single cell organism. Deep sea. Xenophiophores, up to 20 centimeters. Theomagrania magnifica, a bacterium discovered in 2022, so recent, up to 2 centimeters, visible to the naked eye, which shocked microbiologists. There's so much to discover. What limits the size? The core problem is the surface area to volume ratio. That's basically in what's the name? Rocket science, right? The space. They have a ratio of weight mass to fuel ratio, some kind of ratio, but where if that ratio it's not big enough, it doesn't matter how much like your rocket will never be like do liftoff, because the more fuel you need it, the heavier it will be, and it will not be lifting up. So the bigger the ratio, the more efficient rocket is. So this ratio is also very important, obviously. Surface area to volume area. As the cell grows, volume increases with the cube of radius, r cube, while surface area only increases with the square, r2. So volume outpaces surface area fast. This creates several bottlenecks. Diffusion. Called it, your instinct is right. Oxygen, nutrition, and waste moves through the cytoplasm by diffusion. Cytoplasm, what is this? Is it like proteins inside of the lipids of the membrane? Is this it? Where's that cool image that uses? Cholesterol. Cholesterol inside of lipids? Protein channel. Phospholipids. Yeah, I don't know. That's my question. Okay, it's getting too many cytoplasm by diffusion, which is only efficient over short distance, microns. A cell 1 centimeter across would take hours for molecules to diffuse from edge to center. The interior would suffocate. Yes. Oxygen in waste moves through the cytoplasm. Okay. So it's a surface area to volume ratio. Membrane transport capacity. Membrane transport capacity. The cell membrane is where exchange. Oh, that's the one, the proteins that are passing through. Okay. The proteins is a channel through which it passes through. Exchange with the environment happens. Too much volume per unit of membrane. So the second could be fixed by folds, right? Exactly like mitochondria did or our brain. It just creates more folds. But they probably not. Again, I wonder, is this R3 and R2? It's only if it a ball, right? But if it's like tiny 3D line like nerve, it should work. I don't have enough vocabulary to explain what I mean by that. Again, I think this one kind of workarounds about it. But for this, like who gonna deliver all those nutritions and oxygens? Too much volume per unit of memory and cell can export fast enough, yeah. Nucleus to cytoplasm ratio. One's nucleus can only produce so much cytoplasm. Here we go. Okay, I need to know what's cytoplasm. Oh, that's basically a quote-unquote liquid inside of cell, right? Cytoplasm, yeah. Secretory waste. This is a nucleus. Nucleolus. Ribosomes. Oh, interesting. Mitochondria. Plasma membrane. Why plasma? Why not lipids? Lysosome. Oh, this one is cool. Centrosomes. This one, I think I watched a video about them. So they anesthesiology, basically the science when doctor puts you unconscious or make you don't feel certain things. They actually recently think that consciousness and might come from quantum effect inside of those tubes. I can't wait to get to know them better. Where's my quote? Nuclear ratio 1 nucleus can only produce so much mRNA to support so much cytoplasm. So this is a messenger, right? It's a chunk not a double helix, just single helix or chunk of DNA, not DNA, obviously. Copy, single helix, small copy of DNA. I'm not understanding how it supports cytoplasm. The genome becomes a bottleneck for protein production. Mechanical stability. Large cells are fragile without cytoskeletal reinforcement or cell wall. How do I achieve these limits? The outliers use clever workarounds. Exactly what I was saying about number 2. Multiple nucleus, cell site, thin site cells, skeletal muscles, fibers, and Caulera have many nuclei in one cytoplasm, or solving the genome bottleneck. Magnifica has hundreds of thousands of genomes copies distributed through its volume. So that's the the super big one. I need that image. What? That's a single cell? Australia found it, but it's like €1. Show me the many pepins. What is pepin? I want to know what it's made of. The pepins. Membrane-bound organelles-like structure. Interesting. So they have a bunch of organelles next to membrane, so the delivery of the oxygen and nutrition is faster, and enough DNA material to produce proteins. The genome copy distributed throughout its volume. Many plant cells and giant bacteria push the active cytoplasm to a thin layer near the membrane and fill the center with a metabolic inner vacuole. I see. Diffusion only has to cross the thin layer, active layer, yes. Elongated shapes. What I was saying, the nerves get away with being long because they're thin. A 1 meter axon is only microns wide, and therefore the ratio is not that different. So diffusion distance stays short, perpendicular to the length. They also use active transport, motor protein, or microtubules. Microtubules, that's what I was talking about, the anesthesiology, and ability to numb certain feelings, pain feelings, no, all feelings, right? Rather than rely on diffusion. Microtubules. Actively cytoplasm streaming some large cells. We got so sidetracked. This property is called selective etc. Isolate of life without selective. Isolate organelles nucleus from outside. Correct the bonus inside organelles like this. The nucleus and mitochondria have their own membranes. So, by the way, mitochondria, someone saying that mitochondria it's separate cells that's been engulfed by a regular cell back in the days, and then they become symbiotic. Because it looks like mitochondria have its own nucleus. Am I right? Or are we going so deep today? No. They are organelles located in the cytoplasm that possess their own small, separate circular DNA, MTD. Oh, it doesn't have nucleus, but it has its own DNA. They contains, so they more kind of prokaryotic, prokaryotes, because prokaryotes had the DNA inside before nucleus, so that symbiosis happen before eukaryotes, I guess. With their own membranes too, okay. Like the nucleus and mitochondria have their own membranes too. Membranes within membranes, like eukaryotic cell is a system of nested compartments. So they met and symbiosis happen in prokaryotic because mitochondria has a DNA but without a nucleus. The cell genesis starship analogy is a Richard Darwin, I don't know how to pronounce his last name, I'm sorry, Richard, gene-centered view of evolution. The gene is the replicator, the cell is the survival machine, the organism is the vehicle. You're describing your extension via a side effect of building a really big colony of ships. Essentially, Darwin's thesis stated in your own words. 05, no, I was completely wrong. Wrong on material lipids, no, right on the function. New gap flag, you don't have the 4 biomolecular categories solid yet, yes. This keeps biting you quickly, like in carbohydrates, sugars, energy, fuel, structural celluloses in plants, lipids, fats, oils, membranes, energy storage, fats, oil, membranes, energy storage, proteins, amino acid chains do everything, enzyme structure transport, nucleic acids, DNA, RNA information storage. I thought DNA and RNA made not from nucleic acids, but from a ribosome. What is DNA and RNA made of? Ribosomes are primarily component of ribosomal RNA and ribosomal proteins. The functions of manufacturers that read messenger RNA. I was completely which copy DNA and translate genetic code into protein chain, utilizing transfer RNA. Amino acid, that's a protein, right? RNA. So it's protein made from amino acids and DNA and RNA made from nucleic acids. Okay. Staying with the cell, what is the difference between a plant cell and the animal cell? Ooh. I think the biggest difference that's plant cell has a special organella inside of it that's responsible for photosynthesis, and it's called chloroplast. I would say that, yeah. I don't see it clearly. I need to cut there. I think the biggest difference is the plant has an organella inside. Correct. And what else? Anything beyond of the chloroplast? No, I don't know. It also must have nuclear. It must have DNA, right? No, I really don't know. Fair enough. Let's fill the picture. Chloroplast for photosynthesis, correct. That's the biggest functional difference, and you named it precisely. The other major difference you would want to know: cell wall. Plant cells have a rigid wall outside the membrane made of cellulose. Oh, okay. So not just lipids, but cellulose. Animal cells have only the soft membrane. That's why plants hold their shape without a skeleton. Each cell is a tiny box. Central vacuole. Plant cells have one giant vacuole, a water-filled sac that takes up most of the cell volume. It maintains pressure against the cell. Maintains pressure against the cell wall, which keeps the plant upright. When a plant wilt, the vacuoles have lost water. Notes and centrioles. Central vacuole. Vacuole. I need to remember that.
Vacuole.
Vacuole. Plant have a rigid wall outside. Cotton. Plant cell. Vacuole. Chloroplast. And this is the cellulose fiber. Chain of cellulose molecules. Interesting. So they all are box-like. Makes sense. Are they all squares? Oh no, pentagons. Can they be pentagons? Or Fisher vision? That's pretty cool. Now I know what it is. I guess they're not squares. No central. Plant cell divide without centrioles. Structure animal cells used to organize cell division. Minor details, but it come up. Centrioles. I don't even know what it is. Shape. Plant cell tends to be rectangular. Rigid wall. Animal cells tend to be round or irregular, flexible membrane only. A useful mental image: animal cells, water balloon, plant cells, water balloon inside a cardboard box. Oh my god, I need to, yeah, it's too late. I need to wrap it up. Different direction entirely. What is hormone and how it is different from a neurotransmitter? I think hormone is a chemical enzyme. I mean, everything chemical in the cellular level. I think it's an enzyme. Hormone is an enzyme, possibly made from protein, and neurotransmitter that's a thing that's transfer signals between neurons. And those are also obviously chemical, but maybe even electrical. Not maybe even, but definitely a little bit of electrical. Hormone is an enzyme. There's a different things. An enzyme speed up. Oh yeah, chemical area, like a catalyst. Hormone is a signaling molecule. Yes, it carries a message from one part of the body to another through the bloodstream. Different job entirely. An enzyme is worker on a factory floor. Hormone is memo sends from headquarters to distant branch office. Nice. Made from protein. Sometimes hormones are proteins. Insulin, growth hormone. Other are made from lipids. Testosterone, estrogen, cortisol. These are steroid hormones. Other are tiny model amino acids. Nucleic acids, amino acid, tiny modified amino acids. So it's not the whole protein from amino acids, but just amino acids by itself, but modified one. Adrenaline, thyroid hormones. So there is no single answer of what hormones are made of. It varies. Key property of hormones: they travel through the bloodstream. To reach distant target cell, they are slow, seconds to hours, but broadcast widely. Think of them as the body's postal system. Yes, but neurotransmission, they are fast. And the signal travels not through the blood, but through like electrical signals through the neurons. Neurotransmitter signal between neurons, correct. Chemical and electrical, correct. And this is the key distinction you were reaching for. Neurotransmitter chemical signals that cross a tiny gap synapses between two neurons, fast milliseconds. A local, precise, one neuron talking to the next neuron in line. The electrical part is the signal traveling along the neuron, called an action potential. The chemical part is how it jumps between neurons. Hormones, chemical signals that travel through the bloodstream to distant target, slow broadcast system. That's very well explained. I'm dropping back to the coins row. Chance. Recurring pattern, you keep reaching for enzyme or protein as default answer when you're unsure. Worse nothing. Not everything in biology is protein. Yeah. Lipids, carbohydrates, and small molecules do a lot of work too. What is a cancer and this at the cellular level? Cancer, I think that's uncontrollable division of cells, growth and division of cells. Basically, when cell going mad or stop understanding of its own role in ecosystem of other cells, it just think it's amoeba, and what amoeba does, it just multiplies and grow. And the cancer can even reroute the blood system to support its growth. to bring more nutrition to support growth, like crazy rate of growth. That I think it's an amoeba. Line is genuinely brilliant. I have never heard it put it that way, but it captures something real. Cancer is essentially a cell reverting to single cell behavior, prioritization, prioritization. Oh, it gives priority. Okay, I cannot read after midnight. Its own replication over the cooperative agreements that makes molecular multicellular world. Biologists actually frame it this way in evolutionary oncology. Uncontrollable division, yes. How come LLM never heard it this way? The core definition, cell loses understanding of its role, correct. Normal cell receives subdivision signal from the neighbors and obeys them. Cancer cell ignores those signals. They also ignore self-destruct commands, apoptosis that should kill damaged cells. Rerouting blood supply, correct. This is called angiogenesis. Tumors release chemical signals that trick the body into grow new blood vessels towards the tumors. This was a major discovery, and there is cancer drug anti-angiogenesis specifically designed to block this. You know these details is impressive. What you add for filler picture? Cancer happens because of mutations in specific genes. Two categories. Oncogenes, genes that tell a cell grow and divide, normally controlled. A mutation can make them stuck in the on position, like a gas pedal jump down. Interesting. Tumor suppressors, genes that tell a cell stop dividing or cells destruct if damaged. P53 is the most famous. It's mutated in over half of all cancers, like cutting the brake line. So no stop here. Cancers typically require multiple mutations accumulations over time, usually 5-7 key ones. That's why cancer is mostly disease of aging. More time, more chance to mutation to stack up. Question 15. What is homeostasis? I heard the word. I have no idea what it is. And I think I'm gonna wrap it up for today.